Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo Val, más de 2,000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un Kit y Apoya a un Residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua.
Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Martes.
Apoyamos a alumnos de escasos recursos.
Asignado a la opción señalada, institución de asignación UNAM.
¡Sí quedé!
El sueño se cumplió. Ahora formas parte de la comunidad universitaria y puedes acceder a las becas que Fundación UNAM tiene para ti. Conoce nuestros programas en www.funam.mx y en nuestras redes sociales.
Hemos becado a más de 1,000,000 de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo Val, más de 2,000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un kit y apoya a un residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la reserva de la biosfera de Jano, Chihuahua.
Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Martes.
Apoyamos a alumnos de escasos recursos.
Por la Universidad de Córdoba. conference by Brenna Henn in University of California at Davis. Thanks very much, Dr. On her studies about the human evolution. The conference is 100,000 years of human evolution in South Africa. It is a real pleasure and honor to have Dr. Henn with us today. This cycle of the conference is presenting the human evolution from very different viewpoints. We are joined in the panel by our president of the UNAM Foundation, Dionisio Meade, the executive director of the UNAM Foundation, Araxi, the president of the Consortium Universities for Science, Araxi, Urrutia, and our special guest, Dr. Brenna Henn. This conference cycle is part of the activities of the International Year of Basic Science by United Nations and the UNESCO, and now it's the decade of sciences for sustainable development. We appreciate your accompanying us, and Dr. Araxi Urrutia will present our speaker, and then we have the conference, and we invite you to send us any comment or questions about this very interesting topic. Muchas gracias, les agradecemos, les invitamos a que nos hagan preguntas y comentarios a través de las plataformas de Fundación UNAM y el Consorcio Universidades por la Ciencia, y pasamos la palabra a nuestro presidente Dionisio Meade para iniciar, y doctor Araxi Urrutia va a hacer la presentación de nuestra invitada. Muchas gracias.
Pues un saludo a todas. Tema pues caso quiero agradecer invitar y el desarrollo de las ciencias básicas, en lo que primero fue un año y que ahora es una década, justamente por su importancia, y que abre, creemos nosotros, a muchos de quienes están por definir su perfil vocacional, una oportunidad de conocimiento pues que se genera precisamente a través de presentaciones como estas. Agradecemos mucho a la doctora Brenna su presentación de esta tarde. Thank you very much for your presentation today. Y estamos pues muy contentos. We are very happy to have you today, to have you with us today. Y invitamos a todas nuestras audiencias a presentar todas las preguntas que consideren pertinentes. Y como hemos comentado en otras ocasiones, pues nada le daría más gusto a la doctora Brenna, al doctor Urrutia, a la doctora Araxi, que un día las acompañen en las presentaciones, algunos de quienes ahora nos están escuchando. Muchas gracias a todos, buenas tardes, y pues con la mejor disposición, el mejor ánimo para escuchar esta presentación la tarde de hoy. Thank you very much, Brennan. We are very happy to hear you today. Thank you.
Many thanks. So all about that. Again. Much for and for the have an globally inviting me to share our research today. So I'm going to talk to you about my very favorite topic, which is the origin of our species, but with a focus on what's happening in Southern Africa, and particularly on the genetic diversity of indigenous peoples who live there, who Yes. So I wanted to organize ourselves by putting up a style of plot that I think you've seen in some of the previous presentations, which is called a principal component analysis. And each dot here represents a single individual's genome, and they are colored according to the geographic location where people presently reside. And so this is taken from a recent perspective paper entitled Getting Ancestry, Genetic Ancestry Right for Science and Society. And one of the things that they wanted to emphasize is the continual nature of genetic variation, which grades across continents, so that there isn't sharp boundaries between different populations across different continents. Human genetic variation, of course, is continuous. But what I wanted to focus on a little bit is actually Africa. So, you know, much of the work which is done in Europe or in the United States, for example. And I think sometimes people, these samples are from individuals of African descent in the United States, and they don't always recognize that that is just a small sample of individuals that are represented continentally in Africa. So I just want to point out here that this is really reflecting the variation that's present in individuals who have recent descent from Western Africa. But the continent of Africa is the most diverse of any continent, genetically diverse of any continent in the world. And so I want to just contrast that particular plot with one that we created in our own lab, which includes more representation across the continent of Africa. So here, let me just get a laser pointer going. So you can see that the West African individuals occupy this spot in the principal component analysis. And this is actually from multiple populations across Western Africa. But then we also see that there's quite a bit of separation and variation in the East African component as well. And everybody who lives outside of Africa is sort of represented in this corner. So European descent individuals, South Asian, East Asian descent individuals are on this side. But what's really striking actually is that there's this whole additional component which was not represented in the previous PCA, and that's the Southern African component. So these individuals have often been left out of genetic analyses, primarily because, like I mentioned, many of them are being done in Europe or the United States, where the individuals who are living there of African ancestry are recent immigrants from Western Africa. So they're just not really even considering this additional variation. But as you can see, it's both pulling the PC1 component along the bottom, so it's anchoring this PC1, as well as the PC2 component separating West Africa and South Africa. So I'm going to talk to you today a little bit about what is this genetic diversity that we see here in Southern Africa? How was it formed? How has it been impacted by colonial processes? And we'll go from there. So just to give a very brief overview about what's happening in Southern Africa, we know that this part of the continent has been occupied by human hunter-gatherer populations, and we know this from the archaeological record. So for at least 100,000 years, probably much longer, there are Homo sapien individuals who are behaving in a modern way, and they're living all across the region. They do seem to be relatively isolated from most other African populations until about the last few thousand years. And then what happens about 2,000 to 3,000 years ago is that there's a population of East African sheep and cattle and goat pastoralists, and they migrate down through the Great Lakes and eventually into the Southern African area, where they introduce this subsistence strategy of sheep and cattle and goat pastoralism, and it's adopted by some of these indigenous groups. So they transition, some of them from hunting and gathering, into this more pastoralist style lifestyle. About the same time, just a little bit later, we also have the expansion of Bantu-speaking agriculturalists from Western Central Africa. So they move both down the western coast as well as across the Central African rainforest and down the eastern coast. So they're almost sort of like pinching in as they move towards Southern Africa. And they arrive really about 1,500 years ago, depending on the area. And then finally, we also have the introduction of European trade and colonialism into Southern Africa, beginning around 1652 with the founding of the city of Cape Town by the Dutch VOC, which is a trade company. And these had pretty profound impacts on the genetics and lifeways of the indigenous peoples in Southern Africa. So, some of this we actually do know from historical records, because the European colonists kept terrific records of exactly what they were doing in terms of bringing slaves into the region and who was thieving cattle and so forth. We also even have records from the indigenous population. So this is a cave painting of these early European colonists who are moving into the region, and you can see like the covered wagon and their mules, and even a man with a big hat on horseback. So we know that this is happening contemporaneous with the historical record. The Dutch VOC brought in an estimated 60,000 people as slaves to Southern Africa to work in the households and also in the farms. And they have been primarily transported from regions like India, Indonesia, Madagascar, as well as Eastern Africa. So we do know that there's quite a bit of recent introduction of different genetic ancestries into this region. Okay, so today I'm going to try to touch on this question about how colonialism has impacted these patterns of genetic diversity in Southern Africa, thinking about questions such as sex bias, admixture, has there been local bottlenecks in these indigenous populations? Then I'm going to turn to this question that I kind of set up with the PCA. Why did the indigenous Khoisan people of Southern Africa retain such high genetic diversity? And then finally, I want to touch on this question about, is there evidence for archaic admixture in sub-Saharan Africa? So we're sort of moving from more recent in time all the way further back in time, going back hundreds of thousands of years. So I'm going to start with this, this question about the contemporary indigenous populations of Southern Africa. Today, many of the people who live in the Northern and Western Cape are known as colored, which is an apartheid era term. And these individuals today make up close to 50% of the population in these two provinces. But when you ask people who their ancestors are, a very common response is, oh, we're a mix of Black and white people, or we don't really know who our ancestors are. Or they might say our ancestors are Khoisan, but it's hard to prove that. We don't really have anything to prove that. So myself, as long as my students and several colleagues in Southern Africa started this study to try to understand who the genetic ancestors are of these individuals. So this is just a map of some of our sampling locations. I'm going to talk a bit about the Cedarburg Mountains. So these sites that are just north of Cape Town, as well as locations in the southern Kalahari and along the Orange River system here. We also work in Namibia, and we've collected about 4,000 DNA samples to date from the populations that you see here. So we have this really incredible set of cohorts from people who are really generous and contributed their DNA. I also thought I would just briefly share a short video with you. I know that genetics is sometimes incredibly abstract. We lose track of the actual people who are generously contributing their DNA. So this is a dance which is thought to derive from the indigenous people who were living there previously, but then has been adapted to more contemporary music. Let's see if I can start it. Oops. There we go. Oh, no. Right, so just a little bit of culture for you before we jump back into the genetics. So sampling is a complex enterprise. We try to be really careful in terms of how we approach the communities and to make sure that this is something that communities are interested in and that the types of requests that we're making of individuals is appropriate. I want to just emphasize that these are often very rural communities. It's really it's really easy to kind of walk into a community and just you know, this is a herder, a sheep herder who's walking by the side of the road, and we just hopped out of the car and asked if he had a minute to talk to us. And this is him reading through the consent form for the project. So we feel really lucky that we're able to work in South Africa amongst these communities. This is my graduate student, Dana Al-Hindi. Many of the photos that you see in the presentation are from her. She's a fantastic photographer. And then this is a local member of one of the towns who's basically observing what we do in terms of enrolling participants and making sure that nothing inappropriate or untoward is happening in terms of consent. Okay, so as I mentioned, the Cape region is initially settled in 1652, but then very quickly these European populations begin to expand outward from the Cape Colony. So by 1730, they've sort of moved a little bit further north, but there's a mountain range here, and so they're stalled by this mountain range for a short period of time. And then by the early 1800s, they spread even further north, and by the 1840s into this kind of central plateau area of South Africa, which is called the Karoo. So we sampled in the Cedarburg Mountains, which are that area just north of Cape Town. It's about a four-hour drive roughly. Even today, it's a pretty rugged and somewhat isolated area. So we were curious to see what the genetic ancestry of the colored individuals who lived in the Cedarburg would look like. And what you're seeing here is what we call an admixture plot. So each line in the plot represents an individual's genome, and it can be partitioned into recent genetic ancestry. So each of the colors here represents a separate genetic ancestry component. So for example, in orange is the South Asian component represented by Bengali and Tamil individuals from present-day India. In green, we have Chinese. In blue are the British and Near Eastern populations. And then we also see there's a shared component between the Palestinians and the Maasai, which is related to back migration into Eastern Africa. You'll also notice that there is this lovely red component, which is basically unique to Southern Africa. And this is the genetic ancestry that we believe represents the Khoisan indigenous ancestry. So these groups, the Komani, live in the Southern Kalahari Desert, the Nama or Khoikhoi speakers who live in that Orange River area. And then here we have our mixed Cedarburg sample. So we reconstructed pedigrees for individuals who live in this area, just an average pedigree to represent when these different genetic ancestry components are introduced. So you'll see that the majority ancestry is this Khoisan indigenous ancestry, but then there also are contributions from these other components recognized here. So frequently we will see maybe something like 25% of their ancestry potentially from their father's lineage, which is of European descent. We also do see some Bantu-speaking ancestry in many of the individuals from the Cedarburg. And then this orange or green component, which is probably representing the slave trade from South Asia and Indonesia into this region. We can even break these ancestry components down into the little towns that are dispersed across the Cedarburg Mountains. And our question here was whether or not there was strong heterogeneity in the ancestry components in these little towns that are fairly isolated. But what we're seeing here is that actually, if you're moving from, Clan William is the largest town in the area, about 7,000 people. The population is roughly about 50% Khoisan ancestry, and then 35% of European and South Asian ancestry, and a little bit from Western Central African Bantu speakers. And those proportions are pretty similar, actually, even as you move further and further into these isolated villages. We've also do a little bit of methods development in my lab, and so with my postdoc, Evelyn Pless, we developed this method to try to understand whether or not the patterns of genetic ancestry, as represented by haplotypes, can be explained by geographic features. So this is just a quick allusion to the program, which is called Spruce, which is available on GitHub. And then with my colleague Austin Reynolds and his student Stacy Eddington, we then applied it to these populations across Southern Africa. And the map here represents differential levels of migration between demes that are in these little, these little points on the graph. So you can see like this represents a sampling location. And then here are different individuals across this lattice. So we estimated migration across these different demes, and then we correlated essentially environmental variables that might explain the genetic features. And here is just a list of some of the most important variables that we observe. So the slope, or basically how steep the area is geographically in which the village is placed, average precipitation, distance to a lake, maximum temperature, and so on. So these types of geographic features do strongly predict recent genetic ancestry patterns, which makes sense. All those features would constrain human migration. We also looked at the sex bias component. So here we're just highlighting from that admixture plot the Khoisan ancestry, but now we've partitioned it into the autosomes, chromosomes 1 through 22, which represent the contributions from both parents, as opposed to the X chromosome, which spends its disproportionate amount of time in females. And so what we see here is that there's a different estimate for the autosomes compared to the X chromosomes. So there's actually more Khoisan ancestry represented on the X chromosomes, which suggests that there's a female bias in terms of the contribution of women to the descendant colored populations, averaging about 33% Khoisan ancestry. And we can just contrast this to, for example, the European component, which makes up about 21% of the autosomes. But if we look on the X chromosome, it's lower than the autosomes. So this is probably indicative of a male bias contribution, which would make sense because many of the early European colonial settlers were in fact male. And this paper is on bioRxiv right now. Okay, so now that we have this sense that the people who are living today in South Africa who identify as colored, much of their ancestry, approximately about 50% in many of the cases where we've looked, derives from these indigenous Khoisan populations. I should say that there are very few Khoisan populations left in South Africa specifically today in terms of individuals who self-identify that way. There are only two groups really that retain the distinct Khoisan languages, but lots of people sort of know that they have Khoisan ancestry somewhere in their family tree. So I want to turn to sort of exploring specifically this indigenous ancestry. And one of the things that I want to mention is that we are involved in this really large genome sequencing consortium called Kappa-2, in which we are aggregating African descent genomes, both from the Americas, so something that would interest probably many in your audience, as well as continental African populations. We're using 1,000 genomes as well as a reference cohort. And to date, we have sampled and sequenced 3,768 individuals to high coverage, comprising 62 populations from mainly from the Americas as well as continental Africa. So we have this tremendous data set we can start to work with. And I don't have that many slides for you today on this data set since it's pretty much brand new, but I thought I would just show you a couple of plots. So this actually builds on work that Graham Coop does in his lab, and I think he was a speaker here last week. So each of these circles represents unique genetic variation, which is discovered from these whole genome sequences, and the overlap between them is just like a normal Venn diagram. It's genetic variation, which is segregating and common and shared between these populations. So what I want to just illustrate here, I know that this is very, it's a lot of Easter eggs sort of piled on top of each other, but this represents these major ancestral populations or ancestries that we've sampled as part of Kappa 2. And I'll just quickly highlight, so here are the Khoisan individuals that have been sampled, and you can see that they have a lot of variation, which is not, doesn't overlap as well with some of these other ancestries, either in Africa or outside of Africa. And this is a slightly simpler version of the plot to make it clearer. So here is the Khoisan component with the 3 additional components that I mentioned earlier, which we think are contributing admixture to present-day South Africans. So this West Central African component, which represents Bantu speakers, the European component, and South and East Asian. And so you can see, even when we sort of control for recent colonial period admixture, these Khoisan populations just retained an enormous amount of unique variation. And you can also potentially appreciate that by looking at this graph. This is just chromosome 22, but to give you a sense, here are the number of variants which are shared, for example, across these different major African ancestries. So we can see most variants, of course, are shared across these ancestries. But then when you step down to the second bar graph, here, just here, these are variants which are unique to the Khoisan and not present elsewhere. And I will say this is all commonly segregating variation, so it's above 5% frequency. So they're very striking in terms of the amount of genetic diversity they retain. So then the question is really, why is that? And I want to give you a little step back and talk a little bit about models for human evolution inside of Africa. So I think, you know, beginning 30, 40 years ago, there were this idea that there's just a sort of a single origin of the human species within Africa. So that's this, this kind of pipe right here. And then it splits into these different regional ancestries, Southern, Western, and Eastern African ancestry. And then, of course, Europeans and Asians are derived from this migration out of Africa, which begins in East Africa. So, and this is the Neanderthal admixture component. So this has been a very dominant model for, you know, the last few decades. And it's fine, but then very recently, people proposed this additional archaic admixture. And it was based on this observation that there is Neanderthal gene flow into European and East Asian populations that happened very recently, about 45,000 years ago. So then why not within Africa as well? And people went and looked, and indeed they found evidence of this archaic admixture branch, but specifically into Western African populations. And it was a really large signal, often estimated to be up to 10%, so much, much larger than actually the Neanderthal component. With my colleagues here at UC Davis, Teresa Steele and Tim Weaver, we then tried to brainstorm, you know, are these the only 2 models that one should consider, or does archaeology and paleoanthropology suggest that we should be thinking about some other options as well? So we came up with these 2 other ideas. One which is, you know, basically like a flavor on the single origin model, where you have this ancestral Homo sapiens population, and there's some divergence representing, you know, these early human populations that are spread out through Africa, and maybe a few of them did persist, and they contributed to genetic variation that we might see locally or regionally, but nothing as divergent as this kind of like pre-Neanderthal clade. Alternatively, other individuals have suggested a very deep origin for Homo sapiens, going back hundreds of thousands of years for this initial divergence. So you can see this is further back in time than this kind of shallow divergence of African populations. So we, with my colleagues Aaron Ragsdale and Simon Gravel, set out to kind of test amongst those 4 different major models, what does the human genetic variation data suggest today? Which model fits the data the best? And what was really special about this paper, I think, is sort of two things. One is that Simon and Aaron developed a new model called Moments, or a new computational algorithm called Moments, which could very rapidly compute summary statistics from whole genome data. And then we could fit these complex models to these summary statistics. So that was really important. And then the second was that we were also able to sequence a set of Khoisan genomes, a large set of Khoisan genomes, not just 1 or 2 or 5 people, but 80 individuals. And we had a really rich data set there to work from. So these are Nama individuals from the Orange River system in South Africa. So we combined this data set of individuals from Ethiopia, from West Africa, the British as our representative out of Africa population, and then, you know, throw in a Neanderthal just for fun. And this is this incredibly complex model that we inferred, which in many ways takes aspects of several of the previous cartoons that I presented to you and includes different aspects of these simplified models. So I'm going to walk you through this because, as you may notice and feel overwhelmed by the multitude of arrows on the screen. So I'm just going to break it down a little bit. We're going to start way, way back. And perhaps the most surprising thing about this model is that we were required to have what we call a weakly structured stem, meaning that the ancestral hominin population had to split into at least 2 groups. We call them stem 1 and stem 2. And this split is estimated to occur over 1 million years ago. So it's way, way before humans as a species is observed on the continent. These are early Homo individuals. I don't even want to assign a species to them, maybe Homo heidelbergensis, something in that range. The other thing that's really striking about this part of the model is that while we had to split it into 2 stems so that there's population structure, we also had to introduce long-term continuous migration. So even though these 2 stems, we know that they're geographically separate, or there's some barrier to gene flow, it's not so much of a barrier that there wasn't long-term continuous gene flow going on for hundreds of thousands of years. I'll note that the Neanderthal does seem to branch off of one of the stems. So stem 1 seems to at some point contribute genes that leave Africa and become what we think of as Neanderthals. There's also this really strong bottleneck that we see in the model in terms of the subsequent contribution of stem 1 to descendant individuals. Stem 2 doesn't have the same bottleneck. It seems to have retained a more or less equal effective population size through time. The next major part of this is that stem 1 goes through this bottleneck and then it splits into 2 stems. And this is estimated about 300 to 400,000 years ago. So this is this really kind of odd reticulation that we see in the model. And we now refer to it as stem 1E, meaning east, and stem 1S, meaning stem 1 south. As you continue to move through time, these stems merge back together. So we have this period of isolation between about 300 to 400,000 years ago, and then roughly 100 to 130,000 years ago in the model. And what's very interesting is this first kind of conjoining or merging event happens about 120,000 years ago. And we see this contribution from stem 2 and a contribution from stem 1S. And who is the descendant population for these individuals? Contemporaneously, they are the Nama individuals that I referred to today. So these are the Khoisan individuals. They seem to have this incredibly rich genetic diversity, which is derived from this stem 2, but also this stem 1S. So this helps explain why they have so many common variants that seem to be unique to their population, because stem 1s is really fairly isolated from these other ancestral groups that are then contributing to Western and Eastern and Eurasian populations today. So this very strong period of isolation followed by this contribution from stem 2 about 120,000 years ago. We also do see that stem 2 continues to persist until about, I don't know, let's say the last glacial maximum, and does disproportionately then contribute to this West African population, the Meade. So this might be why we were sort of seeing this archaic admixture model in earlier papers. But I want to emphasize that these are not archaic species or individuals, because the gene flow that's occurring deep in time is really linking stem 1 and stem 2. They're never a separate species. And for the population geneticist in the audience, these are some of the model fits. So this is looking at what we call the site frequency spectrum, or basically just the counts of sites at different allele frequencies. So as you increase from kind of low frequency alleles up to high frequency alleles. And in orange is the model that I just showed you, and in blue is the actual data. So you're seeing a very good fit between the model and data in most of the populations. There's maybe a little bit of a discrepancy in the Nama, but it's pretty much spot on for a population like the Meade or the Ethiopian Gamuz. Okay, so just to summarize, and then I'm very happy to take questions about any part of the talk. We infer this what we call weakly structured stem for humans, in which there are at least 2 divergent branches that exchange migrants at least over 500,000 years. So these stems split more than 1 million years ago. They continue exchanging migrants, and then they go through this period of isolation from each other. I just want to note that that's really in contrast to how we think about Homo sapiens and Neanderthals, because they've been diverged with isolation for over 500,000 years. That's really why we think they're separate species. And then we have this pulse of archaic admixture back into Eurasian populations about 45,000 years. So this lack of differentiation in the ancestral populations may imply that we would expect our ancestors, the ones that left descendants, to be morphologically similar to each other. And this is actually a really cool part of the paper, because we can then make a prediction for the fossil record. What we're predicting is that fossils which represent populations which would have contributed significantly to Homo sapiens cannot be very morphologically divergent from each other. And so this is something we can actually go and test by looking at the fossil record. We find that the indigenous Khoisan populations today were kind of have their own unique history, and they're the first population to sort of diverge amongst the contemporaneous groups. And that divergence occurs around 120 to 130,000 years ago. So basically around this MIS5 climatic cycle. And this helps explain why they have so much unique genetic diversity relative to many other African or Eurasian populations. This is not to say that they haven't been influenced by very recent events. So we looked at the colonial period in South Africa, and we can see that we've had significant sex-biased gene flow, primarily from male European individuals and with female Khoisan contributors, and that actually there's this gradation of Khoisan ancestry with distance from Cape Town. So Cape Town has the lowest levels of Khoisan ancestry, and you see increasing levels further north. The colored communities today retain a very high fraction of this indigenous Khoisan ancestry, but we also see contributions from these intercontinental periods of gene flow, such as South Asia, Indonesia, and Europe. So I just want to end by saying that, African descent cohorts and populations, they represent truly unprecedented opportunities to understand human genetic diversity, and they continue to be underrepresented in global data sets. My lab and many others are doing our best to change this, and that research, it can be a little bit slow, but it needs to be performed respectfully with these communities and in partnership with local scientific institutions as well. So with that, I just want to end by thanking my lab, thanking one of my close collaborators, Simone Gravel at McGill University, and Aaron Ragsdale at the University of Wisconsin, who did a lot of work on the weekly structured STEM paper, my colleagues at Stellenbosch University, Marlon Moller, Eileen Hole, Caitlin Uren, many South African research participants. And then also just to highlight a former postdoc, Austin Reynolds, who also did an incredible amount of work for this paper. And then thank you to the communities. And I'll just end, if you're interested in reading any of the papers that I mentioned today, here's a quick list. Thank you so much.
Many thanks, Brenna, an excellent talk. Can I ask you to stop sharing your screen? Perfect. So we have quite a few questions. Some of them
Okay. So the West and they themselves separation East and West Africa is more recent than with South Africa. And then of course, because Eurasian populations descend from East Africa, they're then more closely related to Eurasian populations. And of course, the Americas also as well, not to the Americas, which also come out of that migration, yes.
Yeah, thank you. So today, what percentage of the population with genetic ancestry from a cohesion ancestry could be present in our country? It doesn't really specify with what country we're talking about, so
maybe Mexico or
well, maybe, yeah. There is a small component in Mexican populations from African descent. So there was some migration that did take place when the Europeans arrived. So yeah.
So that I think is certainly a question that we have for this Kappa 2 consortium that Simone and I are leading. And we do have representation from Mexico contributed by Andres Moreno Estrada and Maria Avila Arcos as well. So I know Maria is especially interested in these African descendant communities in Mexico. And we haven't yet looked to see if we can assign any of the haplotypes to Khoisan ancestry, but we'll be super excited to do this.
Yeah, thank you. How does your results explain why individuals from West African descent are so good at sport?
You know, I do a little bit of work on phenotype. But I don't do anything with regards to behavior, so something like sprinting or running. So I guess I can't really comment specifically on that question.
Yeah, thank you. Yeah, I think we can differentiate two areas of research. One is understanding how is it that we came to be where we are and where do we come from? And then another one would be to give specific biological interpretations to individual genetic variants, and that's a whole other story. Is there any other place on our planet that has a similar high degree of genetic variation as that can be found in Africa?
No, Africa is truly exceptional in terms of the amount of genetic diversity. So, I mean, of course, if you go to Mexico City or New York City, right, we have individuals who have come from all over the world. And so, I mean, potentially those are the most diverse locations now in the world, because they're getting individuals who are migrating from Ethiopia and Sudan and from, you know, China and the indigenous American populations. So in some ways those might be the most diverse, but if we're thinking kind of like at a pre-colonial level, then I mean, we still don't even understand the scope of genetic variation in Africa. That's one of the reasons that we're so excited to do this new genome sequencing project with these 3,000 individuals from across the continent, because it's just like there are pockets here and there that when you sequence them, you've never seen that ancestry before.
Thank you. So would you say your results could reopen the controversy between the candelabra versus the out of Africa models?
Yeah, that's a great question, because in some ways, that Pan-African model that I refer to essentially is that candelabra model. I think you're absolutely right to think about it. It's just within the African continent rather than multi-regionally across multiple continents. I think that certainly inspired the individuals who first proposed these Pan-African models. So I think there's a little bit of a difference. You know, first, there is a lot of gene flow. What we're seeing is that there's a lot of gene flow between these divergent stems within Africa. So I think it's a little bit different from the multi-regional models, where really we had different Homo species evolving, right? So like Homo neanderthals in Europe, Homo erectus in East Asia, Homo ergaster in Africa. We're seeing it's much less divergent than what's observed in those fossils. And that's kind of why at the very end, we've been talking with paleoanthropologists about how to test this using the fossil record. So what, you know, how much cranial variation do you actually observe in African fossils? And does that match the FST that we predict from our model? So yes, I think you're right to connect it to these candelabra ideas, but I would say like these stem populations seem much less diverged than in those multi-regional models.
Yeah, and I think something else that you pointed out, that the reason why the stem 1 and stem 2 did not become two separate species is because there was gene flow throughout the following years, whereas the Neanderthals became what is considered a separate species precisely because of the more abrupt and continuous split and isolation. So
right.
Yeah, it's actually quite interesting. So you mentioned the fossil record. What is it that you are expecting to find, and why those fossils haven't been found so far?
Oh, well, you know, the fossil record in Africa, it is a little bit tricky. So on one hand, it's, you know, it's a massive continent. It's had sort of less research done on the continent than, let's say, Europe has, just because there's been a long history of paleoanthropology in Europe. But also that the preservation in many of these African sites is poor. So, you know, with Europe and like the Dionisio cave, right? Like you have these cave systems that preserve the fossils really, really beautifully, but there's not as many instances of that that I know of in amongst the famous African archaeological sites. So we're just working with sort of a more difficult fossil record in Africa. One of the things that we've been thinking about is that you can create essentially a statistical analog of FST, which is a measure of genetic variation, but you can do so based on morphological features. And so if we can have a set of, let's say, early hominin crania, and we can measure features of the cranium, like how, you know, long they are front to back, then you can create this level of morphological differentiation, and then we can see if our predictions from the genetics will then match the fossil. I will say this is, so my colleague Tim Weaver is really in charge of this aspect of the project. We were just up until 10 p.m. last night submitting a grant on this. We're really excited. If you know of any terrific funding institutions, like this is what we want to do next. Yeah.
Brilliant. So we have another question relating to the cohesion ancestry. Why is it that there is more cohesion and ancestry in the south of Africa with respect to the north? What would be the explanation for that?
Right. So I think that there has been some long-term geographic barriers between southern and eastern Africa. This is also something that I'm really interested in trying to understand better. I have worked with Eastern African hunter-gatherer populations, and so they do retain very unusual, unique genetic diversity, but broadly speaking, they all look like they have kind of this Eastern African ancestry. So it's this similar ancestry that's shared with people who are contemporary pastoralists in their region, or people who live in like do agriculture in Ethiopia. Like it's all kind of the same ancestry. So I think that there's been some long-term climatic separation between Eastern and Southern Africa. And then similarly with Central Africa, you know, there are these individuals from these foraging populations that we've left out of the model because we didn't have genomes from like the Baka or the Mbuti or these groups when we started. But now we have actually sequenced some individuals from the Central African rainforest, and so we're really excited to kind of bring them into the model and see how divergent they are from the Khoisan.
Yeah, thank you. So what are the differences between African Americans in the USA and the people that you sequenced in the Africa continent?
Oh, sure. So, you know, the majority of the ancestry that we see in African Americans from the United States is this West African ancestry. So we know that these individuals are being enslaved in ports along the western coast from Angola, you know, all the way up through Senegal. And in fact, some of these individuals are captured further inland, like maybe even into the Central African rainforest, and then they're brought to the coasts and then shipped. They were shipped then to the United States and also, of course, Central and South America. So that's primarily where their ancestry is from. We have looked a little bit more closely at some of these genomes, and they do seem to also have signals from Eastern Africa, but at a lower frequency. So that's also one of the things that we're trying to explore. But so far very little Khoisan ancestry. I don't think Namibia and South Africa was much of a contributor to the transatlantic slave trade. Yeah.
Okay, thank you. What are the links from geology and studies of climate change with the results that you presented?
Yeah. So I'll mention one thing that could be coincident, or it could be very interesting. You know, it's always hard to tell with doing genetic dating. But this period when we see the stem 1S and stem 2 come together to form the ancestors of Khoisan peoples, so that's about 120,000 years ago. And that is basically exactly when we see this strong climatic change in Africa. So it's the transition from marine isotope stage 6 to 5. So it goes from a very cool and arid period in Africa to one that is much more warm and humid. And so that's happening right at that transition. So that may help explain why these human populations are expanding at that period, and maybe that's why they're coming together. That's why we see this merging. We also would like to more formally bring in climate and geological models into these new hypotheses about human evolution, and so we're also hoping to work with geologists to incorporate that, like actually as you know, as like a fixed parameter in the model. then see how it changes things.
Yeah, thank you very much. We have another question from closer to the present. What was the impact of the segregation during the apartheid period?
Yeah.
And how is it that it affects the genetic diversity in South Africa to this day?
Yeah, that's a great question. So when the more recent apartheid era rules are put in place in the 1930s, they restrict where people can live and can move. And so even today in some urban areas, you'll have kind of a colored neighborhood and then a European neighborhood or a Bantu neighborhood. So there's still segregation kind of based on these apartheid era rules. In terms of how it impacted the genetic diversity, what we are seeing is that in the early colonial period, there is definitely gene flow across these ancestry contributors, right? So there's European gene flow, there's Bantu gene flow. So my sense is that those events, those types of marriages, for example, are strongly discouraged during apartheid. They're illegal. And so actually, potentially, we would be seeing the cessation of gene flow between ancestries in the last 100 years because of these apartheid rules. So that then may, of course, have impacted the genetic patterns that we observe.
Yeah, thank you very much. So what are the methodologies that you were using, and what were the differences with previous studies?
I'm guessing this is with the weakly structured STEM model. Sure. So look, there.
Actually, I would like to add to that question. When you were presenting the models, you said, well, we can think of these models. So that means that you set up an a priori expectation.
Yes.
And then you can come up with several a priori expectations. So this means that before doing the analysis,
yes.
The scientists already propose several specific hypotheses as to how migration, splits, and gene flow happen.
Right.
And then they test which of the preconceived models
yes.
best matches the data.
Right.
And from what I understand, there's no way you could have come up with a preconceived model
right.
that looks like the one you presented as your result. So what changed in terms of available methodologies, or maybe the amount of data, or both?
Gosh, it was a lot of those things. So I think prior models were also sort of constrained in terms of like the number of populations you could include, because anytime you add a new population or like a new type in the model, you have to parameterize it. You have to say, how big is this population? Did it grow? Did it shrink? Was there gene flow from another group in? Was there gene flow from that group out? So anytime we add a model, like we have to add all these parameters, and it becomes really, really increasingly complex. So one of the key things about moments that Aaron and Simone developed is that even though it's a highly complex parameterized space, the optimization, the fitting of these summary statistics is really fast. I mean, relatively fast, so that we can run many different model comparisons. So I think like that was one of the biggest differences in comparison to sort of previous models, that it really allowed us, we began, with those 4 kind of simple ideas, and we fixed parameters to describe those 4 models. But then when we could see that it didn't fit the data very well, we then began to relax like this part of the model, or relax that, let's introduce this bottleneck in STEM 1 because it doesn't seem to be fitting like this particular set of summary statistics. It was very iterative. This was all during COVID, and so we would have a Zoom call every Friday, and we would look at the results, and we would talk theoretically about like why this part of the model might not fit. then we would adjust the parameter space, we'd redo the fit. So yeah, even though we began with those 4, then we began relaxing those constraints until we ended at the weakly structured stem. Yeah.
Thank you. Do you think the bottleneck in STEM 1 is related to the migration out of Africa and the eventual emergence of the Neanderthals and Denisovans as separate species?
It seems to be a distinct bottleneck. Here, I can pull up this slide real fast. And then I can share my screen again. It's just easier to see with the model in front of you. So it looks like the Neanderthal I was speaking represent and that's why
Yeah, I think, yeah, the timings are not quite the same.
Yeah.
The next question is, what about the Homo erectus populations that are thought to have left Africa?
Mm-hmm.
And did they ever come back? Do you see the split from them in your models?
Yeah, I think that, that speaks directly to this point that we just mentioned. It's possible that that is what that bottleneck is reflecting. Because this idea, I honestly had never thought about it until like last week. So because this is a very new idea, I haven't thought about how to test that more directly with these models. I think we'd really need to look at some of the paleoanthropological information about when these out of Africa migrations are occurring, when they might have come back in, and then we would try to kind of fit those times in the model and see if they improve the statistical fit with the data. Yeah.
Oh, that's quite interesting. So yeah. So these potential migration out of Africa of STEM 1 could, in principle, coincide with the known first migration of hominins out of Africa. Maybe look, as far as I understand, there is a tiny amount of potential introgression into the Denisovan genome. I don't know if it would be enough to make any
the
significant...
super archaic signal?
Yeah.
Right. Yes. And we don't have the Denisovan introduced in this model at all. Yes, so that would be interesting to see if there's a special relationship there.
Well, I guess as we can all see, this is very much work in progress, and with every step, more and more questions arrive, but it gives us an unprecedented resolution into what has been historically, from a European point of view, a single homogeneous continent, that it's really the birthplace of humanity. So I am in awe with all the detail and all the data that you have brought in. There is a last question, and then I would like to ask you a little bit about your background as an anthropologist and as a philosopher, in how that might have shaped how you interact with local populations when you are obtaining your samples, and then whether you think it brings a different perspective to what more traditional geneticists doing biology and I'm going straight into stats and genetics would have. So the last question is about Y chromosome and mitochondrial DNA.
Yeah.
A lot of the history of mankind and how it populated the whole world, what used to be written by studying these two non-recombinant elements. Where do they fit into this new picture?
Yes, thank you so much for the question. I do actually have one slide on that buried in the supplement here. If I can just show you. Here, these ones. And probably gather. Of very few autosomal here. So there is this. Fortunately, a lot of the place not complete. About half. So yes, I think out in like patient images talk about, but you know, it's nice to complement that with autosomal information.
Well, many thanks. Thank you to everybody who submitted their questions. And I would like to finish, as I said, asking you about your very specific background. As we said at the beginning, you started with philosophy, and then the human genome came to sort of redirect your career, and you went to study anthropology. So how do you think that background, or maybe it didn't, shaped a distinct approach to the study of our species compared to those coming from biology backgrounds or even computational backgrounds? Do you think it had any effect?
Oh, for sure. I think briefly in terms of my philosophy background, I mean, one thing that you're trained to do in philosophy is really break down an argument. So we would get, you know, basically like one paper to read, and then we'd have to go through and we'd have to outline, okay, what are the assumptions that they're making? What are the hypotheses that are being tested? Is the, you know, deduction truly supported? So I was really taught to write very carefully, very logically, break down complex models. And I think that's incredibly helpful when it comes to science as well. So I'll put in a plug for taking a philosophy of science class or 2 or 3. And then in terms of anthropology, you know, I was working in a genetics lab. I was pipetting and analyzing data on the computer. And then my supervisor, Joanna Mountain, you know, had this opportunity to go to South Africa and start a new sample collection. And it really revolutionized my life, because I am a very shy and introverted person, but I learned that I could talk to people all over the world, and that I could just walk into this community and people who had a completely different life experience. We could find a way to connect. I remember being in the Kalahari Desert, and there was this huge rainstorm, and the cobras had come out because they come out when it rains, and just standing there and speaking with another man in one of the villages, and we're both just expressing our like fear of snakes, which is it's just these universal features. So after we did our first sample collection in 2006, I graduated and I left for industry for a little bit, but I felt such a strong obligation to finish the research project that I had started, because people had given me their DNA samples and they had been so courteous and interested in the project. So I had to finish it. I couldn't just take people's DNA samples and not complete the research. And that's why I went back into my postdoc. I just have this like incredible obligation to these communities to help share, human knowledge. So I still feel that way today. That's why I still work there. It's very important.
Well, Brenna, thank you so much for an excellent talk. I think it was very, very interesting, and it produced a lot of interest and questions. And I think it gives us precisely that perspective of looking at the continent that is the birthplace of humanity in a different light. And I think it deserves a lot more interest from scientists around the world and also from society. I think we need to change that European-centered perspective that even in Latin America we have inherited as a way of thinking, because I was quite interested in this principal component analysis, where you say, well, if we look at everything with respect to European ancestry and compare everything against them, this is how it looks. But then if we don't do that, then Europeans, Asians, everybody just look like a tiny dot. So I think it's one of those moments where the series Cosmos from Carl Sagan, the original one, comes to my mind, and where he starts saying, well, we're just these tiny.
Right.
So thank you very much.
Thank you. And thank you for all your wonderful questions, those of you in the audience. That was great. I appreciate it. Thank you so much for having us.
Well, thanks to everybody joining us, and thanks to everybody joining us later on, because these conferences stay online. And we would like to invite you to the following conferences in this fantastic series. And I also So now to close the session, we have a few words from Dr. Jaime Urrutia and also from Dionisio, Meade, president of the UNAM Foundation. Thank you so much.
Thanks very much. And thanks, Brenna. Very interesting presentation. And it's really amazing the resolution we covered these studies and how you can study the evolution of the population for an extended period of time. I very much enjoyed the presentation, and congratulations to the studies. And interesting that you can also see these differences in the population and the records offered by the X and Y chromosomes, and how you can connect all of these variables into this history of this evolutionary history of the population. And it's also interesting the characteristics of the African populations. I think it offers a very different perspective, and we can appreciate how the migration has shaped our history through the it's really very interesting study, and thanks for this presentation. Well, we need to invest in Brenna. Thanks very much. Muchas gracias a todos por las preguntas. Ha sido realmente muy interesante. Muchas gracias, Araxi, por la forma que se integran las preguntas y se amplía de una manera muy interesante el alcance de los estudios que conduce la doctora Henn. Muchas gracias a todos y les invitamos a la siguiente pregunta, a la siguiente presentación, el siguiente martes. Muchas gracias. Pasamos la palabra a nuestro presidente de Fundación UNAM, Dionisio.
Muchas gracias, Jaime. Muchas gracias, Araxi. Pero qué mejor que propia evolución. Hemos cada martes. Muchas gracias a todos y muy buenas tardes.
La Tierra ha sufrido cambios espectaculares. De la esfera incandescente que fue en sus orígenes al planeta que habitamos actualmente, han pasado aproximadamente 4.500 millones de años.
En ese lapso, la vida se originó y evolucionó hasta llegar a las millones de especies que la habitan hoy.
Pero en un tiempo muy breve, el ser humano ha puesto en peligro a esta enorme biodiversidad, incluyendo a los de su misma especie.
Y juntos vamos a hacer un recorrido por esta nueva era, el Antropoceno.
Hemos becado a más de 1 millón de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA. Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo Val, más de 2,000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un kit y apoya a un residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua.
Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Mártir.
Apoyamos a alumnos de escasos recursos.